later. I'm going to talk a little bit about and briefly go over just some terminology. So when TM and others talk about lipid analysis, lipidomics analysis, we'll at least have some familiarity with the terms. I'm sure a lot of you actually know this already. It's becoming clear that lipids are the primary metabolite that's in plasma. In fact, in most cells, different kinds of lipids make up a large proportion of all the metabolites that you can measure. And the complexity of lipids are much higher than most of the other metabolite classes. In fact, all the other metabolite classes. And so, what's the definition of a lipid? It's a hydrophobic or amphipathic small molecule, carbon ion based condensations of thioesters or ketoacetal. So basically, they're molecules that have hydroxy groups that are linked up with very hydrophobic ends. Fatty acids, glycerolipids, glycerophospholipids, single lipids, sacharolipids, polyketones. And then you can also have these carbocation-based condensations of isoprene units. So these are prenol units and steroid lipids. So basically, the intermediates building up steroids, steroids themselves, and then products of steroids such as cortisol and things like that can be considered lipid byproducts. You can actually, I was like that one. Anyways, so you can. There's a website and programs and information at Lipid Blast. This is a good place to go if you want to look at mass spectrum. This is in silico created MS libraries for lipid profiling. So this is a way that we can get an idea of what's in there. We do this. TM will tell you how we actually go about doing it. Okay, so now that we know that there's a bunch of different kinds, lipids are the most common kind. It turns out that half of the lipids in the blood are sterile lipids. The glycerophospholipids, phosphorylcholine, things like that, are there. The glycerolipids, which have no phosphate group on, are the third largest group, and the single lipids, the fatty acids, comprise the rest of it. So these are in general. There's other lipids that are not that are in there, but these are the main things that are there. Okay, so here's the the nomenclature and the approximate number of compounds. Things are getting much more complicated. There's certain branched lipids that are being identified that have biological properties and things like that. So the lipidome is it's complex. There's lots of them. And because the lipids, especially those with the fatty acids, can have different chain lengths, different double bonds, and so it gets very complex. But this just gives you an idea that there's a couple 100,000 at least lipids that one could potentially measure in biological samples. All right, so again, talked about this. Fatty acids, glycerolipids, these sacharolipids, polyketones. And then the isoprene units, these are the sort of the bonds that you see. So the ketoacyl units or these isoprene units that are part of the cholesterol biosynthetic pathways. All right, a fatty acid. This is a no-brainer. If anybody's taking biochemistry, palmitic acid is one of the main fatty acids in plasma, and it has 16 carbons, and this carbon group down, group down on the carboxylic acid group on the end, right? And then if you hook it up as an ester to the glycerol backbone, the first reaction that usually happens biologically is in the SN1 position, and so you get that. If you don't have the double bond here, then you have ether lipids. And those are becoming interesting from a biological standpoint as well. So palmitic acid, stearic acid, 18 carbons, oleic acid, 18 carbons, one double bond, 18 carbons with two double bonds with C9, C12 is the essential fatty acid, linoleic acid. these are, omega-3 or omega-6 fatty acids. The omega, counting from the omega or the terminal here is why you get, so 1, 2, 3, 4, 5, 6. That's the omega-6. If the bond was here, it'd be an omega-3 fatty acid, and you can't make those. All right. So triglycerides, 3, no-brainer. 3 molecules of esterified fatty acid side chains. And again, because there's 3, there's a high complexity of these as far as chain length and the number of double bonds that you can find in them. The glycerol phospholipids, here's the primary ones. There's some other ones that are lurking about, but these are the ones that we can measure. Some of phosphatidylcholine, so there's a choline group or an ethanolamine group at the 3 position that's separated from the glycerol by this phosphate. So serines, phosphatidylserines, the glycerol groups, phosphatidylinositol, and phosphatidic acid, which is just the acid of the phosphate. And so this is an intermediate of triglyceride synthesis. All these others are derivatives of the phosphatidic acid for the most part, or derived from when you hydrolyze a phosphatidylcholine, you can add these end units on when you do the hydrolysis. This is the distribution of the glycerol phospholipids. I won't go through each one of these, but phosphatidylcholine is the major phospholipid of cell membrane. So 40 to 50% of the lipids in the cell membrane are PCs. PEs are the second most common, or the common, and PEs are enriched in the inner mitochondrial membrane. So these different phospholipids that you can find in cells, but also you can find in plasma, have their distribution is asymmetric in some membranes. The distribution is different in subcellular organelles. And if you grind up tissues, you don't know where they're coming from, but you can isolate mitochondria and get a good idea of what kind of phospholipids are in mitochondria. In mitochondria, cardiolipin is very important. It's a glycerol lipid that has 4 side chains on it. And depending upon how many saturated or unsaturated fatty acids that you have in the mitochondrial membranes in the cardiolipins, it can affect the efficiency of the metabolism that goes on in the mitochondria. So it just gives you an idea of the kinds of things, the kinds of lipids that you can see in different compartments within the cell. Plasmalogens have a hydrocarbon chain linked to the glycerol to C1. So here's the SN1. So you have, instead of a double bond, it's this vinyl ether that's formed here. So it's an ether linkage here with the double bond right after the oxygen. These are synthesized mainly in the peroxisomes. In human heart tissue, 30 to 40% of the choline glycerol lipids are actually plasmalogens. In the brain, about 30% of them, up to 70% of myelin sheath ethanolamine glycerol phospholipids are of this form. And so they're used in specific tissues in specific ways. And so, and you can actually measure these as distinct entities. And again, depending upon the number of double bonds in the side chains, it can have different functions and different physiochemical properties. The single lipids really are formed in a stepwise manner from syngosine to ceramides, to single myelin to cerebrosides to gangliosides. This is sort of the adding different groups and adding additional groups onto each one of these, going from here to getting this choline group off, and you can put an ethanolamine on. It's a single myelin, but it's the ethanolamine form of it. And these compounds are actually parts of the outer leaf of the plasma membrane. They're important for cell recognition and cell signaling. So the ceramides and the syngosine 1-phosphate are known mediators in signaling cascades. They're associated with apoptosis. Ceramides are, quote, bad. They cause inflammation and things like that. And so measuring these things in relative abundance or relative release of these can provide some insight into different biological processes that are going on. the cholesterol esters, acyl-CoA, acyltransferase, ACAT makes these. We all know that several drug companies have been attempting to develop inhibitors of these, some of these enzymes to try and change cholesterol levels. They've been marginal, if at all effective. Bile acids come from after you form cholesterol. As you know, bile acids can be formed primary bile acids are formed in the liver, cholic acid and glycolic acid. These travel into the gut. The bacteria can change these into secondary bile acids, and you can get a good idea of, or you can get some insight, as I showed you earlier on, that some of these have biological properties. Some of the secondary bile acids or primary bile acids can cause sporulation, for instance, of Pseudomonas and help me, diarrhea. Thank you. Yeah. So, and then finally, if you look at the lipid species and lipoprotein particles, when we take a sample plasma, if we actually just take and extract all the lipids out, which is what we usually do, one of the things is that you're extracting these from lipoproteins. And the lipoproteins themselves have different concentrations of different of the phospholipids and glycerol lipids. And so this is just a table in looking at, so in the millimoles per liter in VLDL of the plasma concentration, but then you can actually see that there's a different concentration, HDL2 and HDL1, about the different concentrations of cholesterol esters and things like that. So at times you may want to separate the lipoproteins out to see if you're actually having more accumulation of different phospholipids or other lipid species in one lipoprotein particle than the other. So, so this also gives you some idea of how, this can also give you some idea of how things are going from one place to the other. So we know that, reverse cholesterol transport happens with HDL particles, but depending upon what kind of other phospholipids are in, or what other kinds of lipids are in these particles, the capacity and the efficiency in which these particles are formed and transport these cholesterol to the liver or to the kidney for disposal can be changed. And there's several publications out there now, including, I think, Sue Benathor has been looking at how these things, there's lots of double bonds in the lipids here. You can get oxidation. So some of the oxidation that we see is really because of the changes in the constituent lipids that are in these lipoprotein particles. I mentioned before there's branched hydroxy fatty acids with potential biological activity. Barbara Kahn published this is a review, but a nice science paper that suggested that as you have lots of fatty acids come in, you can actually make these funny-looking branched fatty acids. So there's a hydroxy group at the 9 position that can be now esterified to another fatty acid. And what they've shown is that these fatty acids can activate G-protein coupled receptors and have a biological activity. So changing how insulin signals, changing how beta cells release insulin, changing how much inflammation occurs. And so these kinds of fatty acids, and I think TM we've been talking about for a while now, there you are, setting up assays specifically for this. We haven't done it because we haven't had a lot of demand for it, but if people are interested, we can start working out, trying to figure out how we can quantify or at least get relative quantifications of these different kinds of fatty acids that may have these biological effects. Now we know a lot of G-protein coupled receptors have affinity for different kinds of fatty acids in the blood, some of them more complex, some of them simpler fatty acids or simply fatty acids. And different chain lengths of fatty acids and different desaturations have differential ability to activate a lot of these G-protein coupled receptors. And then I put some slides at the end for reference. It's just a a lot of detail about different lipids and things like that. So I just wanted to give you an overview, so at least, you know, when we talk about single lipids and ceramides and stuff like that, that you'll have an idea of what TM is going to talk about. Happy to take any questions. All right, great. All right, so TM, now you can tell them what you really need to learn.